And greetings, everybody. I hope you can hear me. Welcome to the Evidence and Reasons for the Christian Faith video channel. And thank you to all that are joining us now, who just attended the webinar that has been much anticipated. Thank you to all that are joining us now, who just attended. Sorry, I got a little bit of echo feedback from the delay on YouTube, so I fixed that. So John Maddox, Patrick Alexander, you've been sent invitations. If you'd like them to join the stream, you're more than welcome to. Although I got to tell you, that show is going to be a hard act to follow. So this is more or less just a celebration for a lot of the hard work. Dr. Deweese has worked on the topoisomerase enzyme for 20 years, about 20 years of his professional life. You could tell how knowledgeable he was on the topic. So for some of those who might be joining via recording, and I anticipate there might even be some Google hits that will go to this video. What does this have to do with the evidence for the Christian faith? It is evidence that there is a creator, that Darwinian evolution cannot make these things. The process of abiogenesis cannot make these things. So this, Ben Rex says, I haven't watched what this after show is about. It is about the show, the Zoom call we were just in, and the video. So I sent out some invitations for anyone that, for a select number of people that are not camera shy and who've been on the internet, and I invited them to this after show. If they can't come, I'll just do a solo show. So what do we talk about? Oh goodness. I was talking about the relevance to the Christian faith. So the term, in fact, I don't think creationism was mentioned once here in the webinar with Dr. Deweese, but he is a creationist and a young earth creationist. So the Discovery Institute is very, very, it's been kind of an uneasy relationship with creationism. If you believe in creationism, either young or old earth, you automatically believe in intelligent design. But if you believe in intelligent design, doesn't mean you believe in creationism. Michael Behe is probably the foremost example. And we're joined here by Cindy Lincoln. Thanks for joining.
Hi,
otherwise,
Sal.
it's a pretty boring monologue. So were you able to attend the webinar?
Yes. And I tried
Oh,
to answer
happy
some
birthday
questions.
to your great-great-grandfather, by the way.
Yeah.
Abraham
Better than
Lincoln.
Darwin's Day.
Yeah, happy Abraham Lincoln Day. So I interrupted you. Please go on. You were saying.
I forgot. Oh, I tried to ask some questions during the webinar, and they didn't. I don't think it came through. That was amazing. So if these topoisomerases are in every life form, even the simplest life forms like bacteria and the things they find in the Precambrian, that means
cellular life. Just to be, in case you get criticized, say cellular life. Cellular life. Oh my goodness, we're joined by John Maddox.
What's up, Sal? How you doing, brother? And
hey, what's up? I'm sorry?
I said, how's my audio coming through? I'm actually in my car on my phone. Enough.
Oh, it's coming through fine. So okay. This is a great celebration. And let me just offer a brief prayer. Lord, thank you for Dr. Deweese and the 20 years he's worked. And we live in a troubled world. We may be the rumors of war on the other side of the planet. And God, thank you that we have your reassurance that you're there and you're the designer and creator Let's be with our conversation today in Jesus' name. Amen. Thank you for joining, guys. It's really great to see you all, especially you, John. It's been maybe a year since you've come on the channel, so it's nice to see you again. And thanks for advertising my channel.
I don't know if it's been a year since I've been on. I know I've been in the side chat at least, and a lot sooner than that. We've had combos,
Yeah.
but no, I didn't get to see the entirety of the webinar. I was very irritated myself. I forgot about Eastern versus Central time that I'm on, but I've seen obviously a lot of the stuff that you've shown about topoisomerase and read papers on it and such. It's quite a fascinating little gizmo that's necessary for life to exist, and yet another one of those cart before the horse, chicken and egg problems that seems to plague all things biology. And it really just continues to amaze me. The more that we discover on these things, the more the publications come out, the better our technology enables us to really gain greater and greater levels of understanding of how these things actually operate. The more and more the implausibility of them somehow coming to existence without an intelligent agent being necessary gets, I don't know. For me, it's like we've jumped the shark on incredulity at this point.
Yeah. The reason that I hope they released the webinar, Dr. Deweese is so articulate and knowledgeable. I was just getting blown away, and I was commenting in the side chat. I've worked with his lab for 4 years, and there was stuff I just like, Joe, this is news to me. He's just going over because we get so deep in the weeds, we don't see the forest through the trees. And we're very, each of the researchers is so specialized in his field. And he was just describing things about topoisomerase. I just totally didn't. This is the first time I heard it in 4 years.
It always amazes me that I don't mean this like from a negative perspective. I think it's from a lot of scientists are not into marketing logic. Like why you wouldn't have a presentation like that be live streamed, not just through Zoom, but like through platforms like YouTube, Rumble, Facebook Live, etc. To get much greater opportunity for the average person to, even if they might not understand it all, get access to it. The reason I say that is like at the very end. Again, I'm not saying this to bash. Oh, hang on.
All right, Cindy, go ahead. Looks like John's tied up. So you're asking about the Cambrian? Yes, it would have topoisomerase as far as we know.
What I was saying is if the topoisomerase, it's kind of what John was just saying, the chicken before the egg, and that was brought up in the webinar. But if this is present in all cellular life forms, even the ones that they say are primitive.
Oh, we lost Cindy. Oh, Lord, please keep her safe. I hope it's just a button press and not one of those henchmen from where. So just completing the thoughts, LPP was talking about marketing. These scientists are very specialized. They're really not, we got 2 LPPs. All right, we got Cindy back.
2 down.
Yeah, so I'll just complete LPP's thought. These scientists, they especially like John Stanford, extremely reclusive, and some of them really don't want the public eye. And I have invited Dr. Deweese here, but he's really busy. He writes me 2 or 3 times a day, and so do his associates, because we're working on a paper. And even though I have that relationship with him, his time this is like asking an executive for time, because he supervises so many people and he has so many interactions. It's just really difficult. And this is a little bit sensitive, but the Discovery Institute probably would not want him appearing too much on the creationist circuit. And because the Discovery Institute is promoting his work, I'm sorry.
I wonder if that's why my questions were blocked.
Probably.
But if I could finish that. Sorry, babe. Never mind. Go ahead. I wanted to just
No, no.
finish my sentence that got cut off, and that
Oh,
is
go ahead.
if this topoisomerase is in every primitive life form, then there is no primitive life form. It's just a smaller life form, but it's complex from the get-go. And he used the word coordinated. It has to be coordinated with other things going on. There's just no way this doesn't have a designer, a really, 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 really infinitely smart designer.
Yeah.
And they also brought up the point about it doesn't just require a designer who designs it and then lets it go. It's got to have a master CPU.
Well, there's a lot of, there's a lot of this that we don't understand. Actually, the architecture of the cell.
What powers these things?
Yeah, the architecture of the cell is what we would consider distributed processing. There's no master CPU. It's the whole cell. That's basically the computation. It's for the computer savvy out there, it'd be like Bitcoin. There are multiple computers, but there's no centralized authority, which is incredible. The importance for that sort of architecture is that it can sustain a lot of damage and heal. So anytime you have self-healing systems like Bitcoin, you don't want a central point. You want to at least distribute some of the central processing so that you can have places that could fail and then you could reheal or rebuild.
So the individual elements like topoisomerase or like anything else inside this system, they are operating independently without a CPU, and yet they are coordinating perfectly to do the same.
Yes, it's kind of like traffic.
Task.
I mean, you have start and stop lights, but you know, when you're driving out there on the open highway, the individual actors kind of negotiate among themselves. At least you hope they cooperate, and it's kind of like that in the cell. Each actor abides by certain rules, and they're able to cooperate because everything's just bumping around there in close quarters. Let me just address this. Why is topoisomerase important in terms of God? We want to find on this Darwin day, and Casey Luskin quoted him. Darwin said, if it could be found that there is one organ that could not be built in slight successive modifications, my theory would utterly collapse. You only need to find one. And I've said, don't use the irreducibly complex argument. Just find systems. And topoisomerase is really good because there are not a lot of parts that can be co-opted from other parts. It's life critical. It would collapse Darwin's theory. Whether you make this an abiogenesis thing or an evolution thing, it's apparent that this either evolving it after the first life or before the first life, it's going to require a miracle. And if there's a miracle, there must be a miracle maker.
Right.
And welcome back, LPP.
That he quoted from Darwin.
Huh?
He quoted another quote before that from Darwin that was really good. Like that he was open to hearing both sides or something like that.
Yeah. He would have celebrated if you're really interested in both sides. So, LPP, are you back?
Yes, I figured out what was going on. I had forgotten to click open in browser. I'd opened it in my email app, and that's why I kept having issues. But yeah, in line with what you guys are kind of what you've been talking about, you know, in terms of the signaling and such, you know, I think, and I don't know if you got a chance to see my interview with Ben Galt on the mesh code and the Talon protein. But in relation to things like we were talking about in terms like the Bitcoin blockchain type computation, the functions of the Talon proteins are very significant in that train of thought from a processing perspective. You know, you've got all these different additional functional switches or binary switches that are being observed in real time to be coordinating and sending signals into the nucleus for different protein synthesis and gene expression. It's like we're seeing all this happen, but it's happening, and this goes to Cindy's point about the have we had the topoisomerase basically since the beginning? Well, the same logic applies to the talon protein. It goes all the way back to the most simplest, quote unquote, simplest form of life. And we're observing that it's now it appears to be a necessary component, if you will. So we're looking at all this computation, we're looking at the mechanical function, we're looking at the information required for these things to actually operate and be expressed and executed. And then you also have to have the forward-thinking aspect of how do you have all the type of processing.
Oh, I think LPP's phone just rang, so he had to pick up, probably an important call. So let me just show some of Joe Deweese's publications, and I mean he's first rate. So I'll show. There is Joe in the prestigious scientific journal Nature. This is a secular publication. So he's a creationist, young Earth creationist. He's publishing in a premier scientific journal. So I just want to point that out when they say you can't, you're going to fail if you're a creationist, you reject Darwinian evolution. Joe is a great counterexample to that. And anyone who saw the webinar were just like, oh my goodness, he really is a very sharp scientist. And so he never missed a beat just because he rejects evolutionary theory. And so my point is, on this Darwin Day, what use is it? Except to promote evolutionism. It doesn't help us necessarily do our science any better.
Sal, Joe actually made a point about that. He said that since he looked at things as though if they're different, it's for a reason, rather than coming at it from an evolutionary standpoint. If he's coming at it from the standpoint of, okay, this was designed for a reason, this was engineered for a reason, that it impacts your science and that you can do better science than if you have an evolutionary mindset, is what he was alluding to.
Yeah, and that's about as far as I think. Just to go back a little bit. There's always been kind of a strained relationship between the Discovery Institute and the creationist crowd, and that's also one reason I didn't invite Dr. Deweese here, because I wanted to respect what a tough position he's in. And that does relate to like the messaging each group is doing. That being said, this is still a big deal that they had a young Earth creationist at the Discovery Institute doing this talk. Why would they do that? Because he's a really good scientist. It's like, okay, we'll kind of not talk about our differences, because the Discovery Institute's a lot of old Earth people, and a lot of people that believe in universal common ancestry, like Michael Behe. But together we can come and talk the pure design of life and its complexity. So to that end, Utopia Buster says we have to define complexity. I wouldn't be too formal about it, but basically it's the number of things that can go wrong. So, I'd say a stapler has a lot fewer things that can go wrong than, say, a 747 in the functioning of its systems. A VLSI chip that powers your computer, lots of points of failure. So just that's a good enough measure for most of our purposes. And I could show and walk through all the major points of failure with topoisomerase, and they're numerous. And I may do that. Now, Terry Stark asks, what did LPP mean by that scientists should study marketing? What he was talking about is, okay, I'm going to take a swipe at who here, Cindy. Why don't people like Joe Deweese get more of the focus and attention in the creationist world? Why do we have other people like who, Cindy, get the attention? And that's a good question. I think part of it is they're very, very specialized, and most scientists I know of Joe Deweese's caliber are very reclusive. I mean, he's a very nice guy, very personable. Obviously, you have to be if you're going to be a good professor. He's very caring, but he's just not into getting up in front of crowds and like you see these guys, these performers. There are a lot of creationists who are performers. They're not scientists, and some of the stuff they say is just garbage. And it's actually the evolutionists who do a very good job of marketing them because they're so bad. It makes creationists look bad and Christians look bad.
To knock down. A
Yep.
good straw man to knock down.
Yep. So.
Well, and as I've been trying to transition from Kent's style to professional scientist style, it's a good thing I have a lot of education because, man, you have to be able to focus and concentrate in trying to sift through an hour and a half presentation with big, huge words and trying to figure out what are they saying and how does this have to do with God? It's hard. You have to really, I mean, it's not like watching a cartoon.
So I really hope that, Danish debater said 17 minutes behind. We had that. I hope you had seen the Zoom seminar that this is an after show for. If you haven't, I really hope they publish it because you're going to get to hear a young Earth creationist ID proponent who's a professor of biochemistry and who's published in top tier scientific journals. And I'll show you some of them. I mean, that means he actually does research that the secular world values. And this is one example here in the prestigious scientific journal Nature by Joe Deweese. And I'll talk about, there's a publication he and I did. We heard today about the C-terminal, the variable C-terminal domain in his webinar, and I can talk a little bit more about that if I remember. He also talked about some of the current research, and this is one of the current research areas. And we actually did we published in a creationist journal, and this is our publication here. But it's amazing. Most of what we said in the creationist journal and the secular journals were very similar, except we connected the dots, if you might say, in the creationist journal. When we're talking about the complexity of the system, we could talk more freely and say, we believe this points to a creator God. We can't say that in a secular journal. We can just talk a lot about the complexity and then kind of our research that highlights aspects of that complexity.
So the Design Institute that hosted that Zoom, they believe in design, but they don't want to talk about the creator?
That's correct. Get it. Okay, part of the reason is, I'll explain why. There are a lot of people that are not Christians. They're like Jews, for example. They're Muslims. They're Hindus and sympathetic atheists that believe biology is designed. There's actually atheists who believe in intelligent design. That's a very good observation. And some of actually the best intelligent design theory is from the atheists, who they have various theories as to who the designer is. One of them, Fred Hoyle, suggested aliens, and same with kind of like Francis Crick. Yeah, I know you're laughing. Okay.
Because that just pushes out the problem. So you go to a different life form and you say, well, where did that life form come from?
Yes. There is strength Because if even atheists can pick up the design and say this is not an accident, so what they have to do is formulate where the intelligence came from. That's where we'll disagree, but they'll say, no, this is not what you'd expect. So Fred Hoyle was one of those atheists slash agnostics. I think he's more on the atheist side, who said the complexity of the cell, it's like to imagine that it evolved by itself is like supposing a tornado passing through a junkyard could assemble a 747. That's an atheist who said that. That's an atheist who said that, and he argued with a lot of the abiogenesis researchers, and particularly evolutionary biologists. And he did a very good job of, with where he was 40, 50 years ago in engaging them. I mean, his arguments today would be very obsolete, but he was one of the pioneers. So back to your question, does the Discovery Institute, why doesn't it talk about the creator? It's partly because we have the atheists. We have the atheists who invoke extraterrestrials. We have Hindus, Muslims, Christians of a variety of faith, and Jews. And even within Christianity, you have all the denominations. And the one common thread is intelligent design. The identity of the designer, that's something, they don't want to, they call it the Big Ten of intelligent design. There is some commonality here. And maybe to give a warfare analogy, in World War II, you had the Russians and the United Kingdom and the United States being the major allies against Germany. But, after Nazi Germany was defeated, Russia and the United States were at war.
We're not allies.
Right. So you can view the Discovery Institute kind of hosting this intelligent design viewpoint. But a lot of the players are allies only on that. They will part ways when we start talking about the identity of the creator. And Christian creation.
Didn't even say Jesus. I did not say Jesus. I just said creator, which I thought would be, well, actually, I don't even think I said that.
Well, they may have seen the name Cindy Lincoln in the.
Yeah.
Casey Luskin knows who you are. Casey Luskin knows who you are. And he.
That's why.
Yeah, I'm sorry. I'm sorry, Cindy.
But I don't think I said anything bad or
radical. You just don't get a break. I know, you just don't get a break. I'm so sorry. Hey, but you can hang out with me, all right? So I know it's not as prestigious as hanging out with Casey, but you can hang out with me. And Casey's my friend. Really serious.
I didn't know who he was, and I looked up who he was, and he's done some great stuff.
Oh yeah.
But I was noticing the difference. Okay, so you told me yesterday from the get-go that we did not agree on all things that the Discovery Institute, but that they did a great job in the design part of the battle. And yet now, today, I'm learning they don't offer us the same respect, where they would say, well, we don't agree with everything about them, but we agree with them on this. I found that strange.
Well, it's just like the World War II alliance, just like what Harry Stark pointed out here, Churchill versus Stalin. They were allies. Hard to believe, huh?
You're right.
And this is the relationship between, we are one of the allies in the intelligent design, and there would be people on the other side who are Hindus and Muslims. And that we're definitely not going to agree on the identity of the designer and his nature. And remember, Christendom fought the Crusades against Islam. Okay, so there's a long history of warfare between these two, and I don't expect it to end. It continues to this day. So in the intelligent design camp, we can agree on the complexity of life. And maybe that's about as far as it goes. But
okay, well, can I read one of the complexity of life comments that didn't get shared? Because I thought it was really cool.
Why don't you share your question too that you put, if you remember it, but go ahead.
Yes. So you know how a wire is oftentimes multiple wires all wrapped up in a plastic sheath?
Yes, insulator. Yep. Ben Rex would appreciate that.
The topoisomerase.
Ben Rex is an electrical engineer. Sorry. Go on.
Okay. So if you cut a wire and then you try and reconnect that wire, although I just realized you just have to twist it together. But okay, so this is what I put. The cut strand, the cut DNA strand, quote, reconnecting, they just glossed over that. But reconnecting a DNA strand is like trying to connect each of those separate wires. They have to touch the one that they came from. It's not like reconnecting a homogeneous thing. Each of the individual strands, each of the individual DNA parts have to be connected to their correct part. So the whole reconnecting thing, the animation made it look like it was just magically simple, when if you think about it, that's huge. Do you get what I'm saying?
Well, first off, you have to position it in the right spot. Now, there's some biochemistry, biophysics stuff that, I mean, your analogy is understandable, but at the biochemical level, stuff behaves differently, and that has to be accounted for. But you do get a rough order, very rough order of magnitude of the difficulty. The wires have to be positioned right. I mean, you could just let them separate. And so the trick is making sure they're in enough proximity. Okay, so.
Well, and you're pointing one finger to one finger, but a DNA strand has multiple rungs, right? So you'd have to point 5 fingers to 5 fingers, and you'd have to get each one. That's what I'm trying to say. Is that right?
You have to do a lot. And remember, it has to be separated when it's cut. It has to be just wide enough to let the other DNA strand come between. And after, so it has to be timed right. So first it's cut, then separated just far enough. It has to wait for the other DNA strand to come through. That's the untangling move. And then after that, it's put together. And yeah, I've asked biochemists, how hard do you think you could build a machine that can do that?
And when you're saying about the strand going down through the slot, down through the cut, okay, the animation makes it look like, oh, well, just gravity is making it go down. But then somebody in the chat said, what makes it go down? Because it wasn't gravity.
Exactly.
How does it know where to go?
Upside down. Exactly. This is why I talk about biophysics. The physicists have to figure out stuff like that. And like when he's talking about, it needs energy to hold it together. And like, as a physics guy, that seemed kind of strange. It's like, I mean, isn't there some sort of bonding and you actually have to, so I don't know all that, but that's what the biophysicist had to work out to make these estimates. And that's the work of the biophysicist, figuring out. And they do a lot of stuff estimating the distance and size. And those are what they call the structural biologists. I mean, this is why Dr. Deweese spent 20 years on this, and he's only one of many researchers that has spent their professional lifetime on just the topoisomerase. In the human, so topoisomerase alpha is only one protein in the genome. They're probably 20,000. And this is only one.
20,000 proteins in the genome.
At least. And if we count what we call the isoforms, the isoforms alternatively spliced and otherwise co-translationally edited, it could be 500,000, 1 million. We don't know. We
that was
absolutely.
kind of my question. So the 20,000 things that you're saying are inside one cell, each and every cell?
Human cells. We have a little bit more genes than bacteria, which is also a little surprising since we're so much more complex. You'd think we'd have proportionally more genes and proteins, and that was the surprising thing. We don't in terms of proteins. We probably have way more regulatory systems, and that's a whole nother topic when talking about combinatorial regulation. But we are more complicated than bacteria, but we actually don't have, it's like we almost have the same, a slightly bigger dictionary of words. If we, if we make words like proteins or genes, but we're certainly more complex. So you might say the bacteria uses a slightly smaller dictionary, but it writes children's books. The human is like very intricate novels, and we have a slightly richer dictionary. Okay, so someone called them Utopia Buster here calls them scissor enzymes. Okay, so now I know what you're talking about. If I miss some of the side chat, I can't multitask real well. So yes.
Sal?
If you have more, yes, go on.
I was trying to get a spatial visualization of what these things are. That's why I said, are they in every cell? Because I know a human has trillions of cells. So does each cell have a topoisomerase in it? So
yes,
or it
yes.
so they live inside the cell.
Yes, we have 100 trillion cells roughly. Each cell has roughly, not completely, the same DNA that every other cell has, and each cell will express a topoisomerase enzyme. There are a few exceptions like erythrocytes, the blood cells, but in general, that's just say that's a very good approximation. All right.
So these little scissor things, there's trillions of them in our body, and they're in every cell in every life.
Maybe quadrillions. I'm not kidding. There could be quadrillions of these. And by the way, there's an estimate that there are quadrillions of chemical reactions happening in your body every second. Every second.
Coordinated. It's not random.
No.
The coordination level is staggering.
Yeah. It is staggering. To quote the Psalms, it's fearfully and wonderfully made. So going back to the point a little bit, I'm glad you're seeing the distinction between intelligent design and creationism. So if you're a creationist, you believe in intelligent design. But if you believe in intelligent design, you're not necessarily a Christian creationist for sure, okay? So that's the distinction. But what they do very well is highlight the complexity of the cells. So just like we're having a conversation now, you probably didn't get that at Dinosaur Adventureland. What the Discovery Institute does, they focus on almost one thing. Their Center for Science and Culture is the complexity of life. They do a few other things like the privileged planet and the fine-tuning of the universe, but they really look at the complexity of life, and they're probably the best organization out there that will highlight the complexity of life. And so they do one thing and do it very, very, very well.
And
go ahead.
Casey Luskin, he was like excited to point out that it did basically undermine evolution.
Yes. So by evolution, they usually mean evolution of complexity, which is different than common descent. So even people that believe we evolved from apes would have to say that the steps there had to have some sort of intelligent input at some point, and they're not very specific. So when we talk about the intelligent design advocates who believe in extraterrestrials, and I think even someone had mentioned this, and I don't know where, that the aliens would have to come back periodically to inject the genome to get stuff to evolve. Okay, All right. So he did not go into details. So I'm just pointing out there are people that believe in intelligent design. It would still accept common ancestry. So what do you call those? Do you call those evolutionists or not? You can call them intelligent design evolutionists. So there are a few of those.
For Joe Deweese to be so successful in his field, surrounded by people who disagree theologically with him, he can't just be shooting off his mouth all the time. He has to be very conservative and restrained and yeah.
Well, I just have to say, I think God was with him, honestly. God prospered his way because we know the other side tried to get me kicked out of graduate school. I wasn't even a PhD student at the time. And I was studying physics, which is far removed from biology. And they were calling professors at my school saying, you got to kick this guy out. I mean, there's a lot of hostility. But what ends up happening is if you're talented and you serve, most professors that are not too ideological, or even if they see that you're talented and you can make a contribution to the curing of cancer, why wouldn't they hire you? See, we talked about topoisomerase. He talked about topoisomerase and it's important to the treatment of cancer. And if I'm a cancer researcher and I have relatives that have, if we had relatives that died of cancer, you don't care about all the dogma stuff. You want to get the job done and heal people. And the reason Joe Deweese became a biochemist is his brother got cancer. And when he was young, he wanted to understand more about it. And so he pursued biochemistry. I'm happy to report, I asked, hey, Joe, when I heard your testimony about why you became a biochemist, you said your brother got cancer. I mean, I had some trepidation asking, what happened to your brother? He said, oh, he's fine now. He's living and prospering. So praise the Lord for that.
Oh, wow. That's unusual.
Yeah. Yeah. So God can use tragedy in people's lives to put them in a certain career path. And that was Joe. So.
But Sal, do you think that for college-age Christians who are wanting to pursue this kind of research, they will need to tone down evangelism for the sake of being able to continue? And if Joe or some of these other creationists want to start broadcasting their findings, that they will probably have to leave and work elsewhere,
or they'll be fired. And I told you about the book by Rod Stadler and Change Tan. Change Tan got fired from University of Missouri. She was a tenured professor in a secular university, a very good one. She's tenured for 11 years. She came out as a young earth creationist. And even though the official documents will mention nothing about her creationism, I think,
So she had reached a level where she had made discoveries that were solid enough to publish, and she had to make a decision. Do I keep this to myself and keep my job, or do I publish this and put my job on the line?
Right.
And so she made a decision for the 10 years prior to that she wasn't ready to come out, and that maybe sometimes, because I know Christians have a lot of pressure to be vocal and bold and evangelize. And yet it seems to me like that is kind of a good reason when sometimes we do need to
be quiet.
Just, yeah.
And we have examples in the New Testament. There were times when the apostles were persecuted and martyred, but there were also times that they had to run. And it's really funny. they got persecuted, and then they fled Jerusalem and spread the gospel that way. So sometimes you just go to other venues. In Change Tan's case, if you're marked as a creationist, then you don't get government grants. That means you're not bringing any more money to the university, except for maybe teaching your classes. And at some point they just will let you go, and they'll say, well, you're not getting any grants. And so, legally speaking, the university could say, well, it's just because you weren't getting any more grants. And then
so tuition is not their primary income stream, is not tuition.
We don't, there are all sorts of things, but there are other things that they could do to fire you, like give you a workload that's just unbearable. And then they'll say, well, you didn't meet
turn the heat up.
Yeah. So
turn the heat up.
That's why I said God prospered Joe Deweese's way. But we're all subject to persecution, and this is in addition just to the ordinary politics that goes on everywhere. Like if someone just doesn't like you because for whatever reason, you could be axed, and there could be favoritism. And sad to say, I've heard where certain researchers would hire their girlfriends, and why they picked one employee over someone else had nothing to do with qualifications. Okay, professional qualifications. So, and academia is becoming a very, very corrupt place. A lot of it has to do with money and prestige, just like a lot of corruption. So it's not just a Christian thing. And I don't know what to tell students today. I'm just like, I don't know that I could navigate. Change Tan wasn't a Christian when she first got into university. And so that probably helped her career, actually.
Yeah.
She came from communist China, came here, became a Christian, went to academia, and then she got fired, just like in communist China, I guess. But let me...
Is she feeling okay? Is she feeling okay? Is she depressed?
She's trusting the Lord.
Yeah.
She's trusting the Lord. So let me show this diagram to relate, since you're asking about ID and creationism. Let me see, share the screen. So this is maybe one way to visualize the relationship of intelligent design and creationism. Young Earth creationism, that is. These are kind of how I felt my views changed over time. The very red, dark red, meant I was kind of with the mainstream. This green color means I'm solidly in the creationist camp in terms of how well I feel I could defend the arguments. And that's when I nearly left the faith in 2002. The pictures of the people here are prestigious scientists. There's Dr. Sanford here. These two guys are Nobel Prize winners, and they believe in intelligent design. There's Michael Behe and Michael Denton. Michael Behe definitely believes intelligent design. Michael Denton's kind of in between, but they both accept universal common ancestry, which you can define as evolution or not, depends on your definition. But now I'm going to just, so intelligent design is kind of is in this area here in the green. Young Earth creationism includes intelligent design, but then has all these things about the fossil record, etc. So I don't know if that was helpful.
Yeah, you're just pointing out there's way more facets
to young Earth creationism.
Yeah.
So the discovery, you could say basically the ICR focuses on all this, all these rows here. The Discovery Institute focuses on those three, and I mark them as green because they're very defensible. I believe at some point, you could see how I felt over time about the defensibility of the argument. And you could see definitely there's a trend. Even by 2000,
All the guys that were in those photos that you just took off the screen, they all had the same progression. They went from red to green. All of them, as far as I can tell.
Okay, so these
Well,
Dr.
no, maybe
Stanford
that's
went
not
from
true.
red to green. So did, Walt Brown was probably kind of in the gray area. These guys are, this one, he was an atheist. He became an old Earth creationist. That's Richard Smalley, won the Nobel Prize in Chemistry. Charles Towns here is not a creationist, but he said intelligent design is obvious. He's talking about the universal level. He won the Nobel Prize in physics for the invention of the laser. So your everyday life is very much affected by lasers, anything with an optical drive or optical communications, fiber optics that, push through internet data across fiber optical wires, Charles Towns. And so these are the elite scientists. They believe that the universe is a miracle. And Richard Smalley especially believes that the first life was a miracle. He's an old Earth creationist. So he would definitely be in, like, say, the two green blobs here. So he got converted. He never became a young Earth creationist, but I expect to see him in heaven. And Charles Towns is an evangelical Christian. Michael B. He's a Catholic.
How many people went from red to green through science, and yet they're still saying that science is against religion. It's just so strange. I guess we can only do what we can do.
That's right. I will mention, I think Reginald Cahill, I don't know his religious views, but he is, his work is influencing me in terms of what they call the distant starlight problem. But let me just show where I think where we are. Even in 2022, I'm seeing more of the areas become green. We still have problems with the age of the solar system and the age of the cosmos. I can't defend, I would have a very hard time saying that the cosmos is young. I can only say that there are problems with the Big Bang model at this point. But you could see there's a trend. Because there's lots of evidence we have today. We couldn't make some of the arguments like the topoisomerase argument you heard today. We couldn't have made that 100 years ago or even in the 70s. We can make it today. And so there are a lot of things that we have today that people did not have in the past, that creationists and ID proponents didn't have access to. So anyway, I hope that this diagram is a little bit helpful to kind of point out where I think, where we were, where we are now, which is 2022, and where I think we might be going in terms of defensibility. So even though I believe that the world is young, including the cosmos, I would not be able to make a good scientific argument to that effect. And I freely admit that. But look, I couldn't have made good scientific arguments 20 years ago, but I just waited for data to come in, and the Lord provided. And I think we're at the point we can definitely give a good showing on some of these areas here. So that's the difference between.
I love how some of it is because of evolutionary research, not just creationists, but they stumble upon the truth themselves.
Exactly. And Tony Maurice, really great to see you. So let me now change my slides, because I did promise some people I'd talk about stuff. And while I'm doing that, Cindy, just speak your heart, because it's going to take me a while to set this up. I promised some people some things in the side chat during the Zoom call, and I'm going to just try to follow through with what I promised.
Okay. I'll just look at the questions that didn't get answered again. One of them is, what is powering topoisomerase? And Joe tried to say it was the yellow stuff on top. I forget what that stuff's called. in their,
ATP, adenosine
yeah.
triphosphate.
But that kind of just pushes the problem out, kind of like the aliens. Okay, then what powers that? I mean, what powers?
Oh no, he was talking specifically. He wasn't talking about design. It's just like what would fuel a car. You need gasoline to fuel a car, and ATP is the gasoline, is the fuel that powers the topoisomerase. It had nothing to do with intelligent design or creationism per se.
Well, in my brain, it just pointed out the obvious.
So when he was talking about powering, it was more like fuel for a car. It wasn't talking about the fact you need a designer to make a car.
Yeah, you need power, you need fuel, you need direction, guidance. You know, where do you cut? How does it know where to cut? What if it accidentally cut a gene? I mean, if it cuts in the wrong place,
it may actually cut a gene. It may, but then it can stitch it back together. That's the trick.
Well, I guess I was just assuming that if it makes a cut in the wrong place, it could do harm.
But it stitches it back together. So it could do harm if it didn't stitch it back together. But because it does, there's no harm. In fact, it's because it cuts it, it enables it to untangle it better, and then it stitches it back together. So it's just kind of like a car just repairing a car. You break it apart and you put it back together better, hopefully.
Well, I know, but the point being that if there's just a bunch of little scissors randomly wandering around cutting without direction, that would be harmful. But the fact
really
that they are just plain cutting the gene strand, they're not cutting the cell wall or each other, or you know what I'm saying? I mean, scissors cut everything. And so they are very precisely pinpointed.
They are very precisely pinpointed. That is correct. And so there was that part in the webinar where Joe Deweese said, yeah, topoisomerase likes to hang out in certain locations in the cell. And he specifically used the word nuclear localization, which I use a lot. You'll hear me talk about nuclear localization. The nuclear localization signal, which I think why don't I show my screen here? We'll talk about nuclear localization. See if
I can. Is that those little gates that go into the nucleus? Or that's a cell? I forget.
That's the nucleus. Exactly. Nuclear localization. So let's go. This is a paper I'm working on with Dr. Deweese, and it's fine for me to talk about it because we might preprint it anyway or whatever. So this is the spelling of the topoisomerase 2 alpha, which he talked about, and there's also the 2 beta. So let me just back up a little bit. So we have amino acids, and we have 20 canonical, and then we have some special ones that aren't listed. And you could see amino acids are made of hydrogen, nitrogen, carbon, and sometimes there's one here with sulfur somewhere. One of them has sulfur. Oh, here it is. Cysteine has sulfur in it. So carbon, oxygen, nitrogen, carbon, oxygen, hydrogen, nitrogen, and sometimes sulfur in the case of cysteine. And to each of these amino acids, we can assign letters. So we use English alphabetic letters, and there are all these properties of the amino acids in this Venn diagram. And the topoisomerase is spelled, you can spell out the sequences of all the amino acids that make the topoisomerase, and it has to be in the right order. You can't just take these letters and randomly put them together and have a topoisomerase. So you saw all the things that topoisomerase can do, and it took an hour to just describe some of it. but it's only, this is shorter than a paragraph in terms of the spelling. This is how cleverly designed it is, that it could be, so much function can be just in a few parts, relatively speaking.
And what you showed on the previous slide is all the amino acids that are spelled out back to back to make that paragraph.
Correct.
Is that right?
So like A is for alanine, R is for arginine, and N is for asparagine. And you could see actually the molecular, like there's A for alanine, V is for valine, I
so
is
to make
for
a
isoleucine.
topoisomerase, you have to have, so right there where you see the N, that stands for an amino
Methionine.
acid.
Okay, so let me see if I
can
can.
Chain in itself.
Right.
It's either a chain.
So E, right.
Oh my gosh.
Okay, so yeah. Okay, so you have each of these is each molecule here's an amino acid. So you have the M, which is methionine. So there's the methionine here.
Where's the N?
N is asparagine. Let me see if I could find it. Arginine. Where's asparagine? Here. This is N right here. And then we have an E, which is I'm embarrassed.
So it's a molecule.
Glutamate.
Essentially, it's a molecule.
Right. So this is the amino group here in the dark black, and then what they call the R group in the red. So you see that the black, it's identical across all the amino acids. So that's the signature right here. You want to have this part that makes it an amino acid. The red part is what makes all the amino acids different.
Okay.
So you can almost think, I mean, it almost looks like a key. So you kind of like to have the key handle here, so to speak. And the way the amino acids are connected to make a polypeptide or protein is, oh boy, I'm going to mess this up. You take the N-terminal part of the amino acid and connect it to the C-terminal. So N, C, like that. And you just connect the N's to the C's. This is the carboxy group here. And this is the, I'm going to blow this. Dr. Carter, where are you?
Just say black and pink. Just say black and pink.
But what you see is the black COO connects to the H3N, and then something is released in the process. I think water. And so you can just string them along, and then you make something that looks like this. So let me read this off. You have the methionine.
So that one little N represents a really complicated molecule or string of molecules. I mean, the whole thing is just
letter is M. Okay, so the first letter is M.
Oh.
And M is methionine, which is here. So it represents just this molecule right here.
Okay.
We call that a monomer. So a monomer, or just call it an amino acid. It's like you might say a bead on a string, but I have to be really careful because I use that analogy a lot for other things. So you take the methionine here. The next one in the spelling is glutamate, which is this with the E. So, and then the next will be valine, which is this. So the first three letters you'll see M-E-V. So you have the M first, then the E, then the V. Yeah, and you have this, and to the right of it, quote unquote, is this. To the right of this is this. And then you can see how it's spelled out here. M-E-V. And the next is serine, P is for proline, L is for leucine, Q is for, and I'm embarrassed, I don't know what Q is. Q is, where's the Q? Glutamine. Glutamine. So methionine, glutamate, valine, serine, proline, leucine, glutamine. So there's a glutamine and then a glutamate. Glutamine is Q, glutamate is E, P is proline, V is valine, N is for asparagine, E is glutamate, N is for asparagine, M is methionine, Q is glutamine, V is valine, N is asparagine, K is for lysine, I is for isoleucine, lysine, asparagine,
So these things
glutamate.
just line up next to each other like little soldiers.
Yes, D is aspartic acid. Yep, right. And then, and there's 1,531 of these in the topoisomerase alpha, which was the subject of the video. So when you have these things all lined up, they make something that looks like this. There are other ways to render this. Let's see if I can.
So they're not lined up in a line. They're making the shape of that topoisomerase.
No, actually they are. It's a string that is shaped. It's a shaped string. It literally is
so
like
it
taking a
knows
necklace.
Somehow when to fold.
Yes. So you take a necklace, you can fold it all sorts of ways. But that's another step that's amazing that it can fold. Okay, so here start. No, this is very educational. See, I keep sometimes forgetting that it's good to spool people up, and at least you have the patience to endure.
Yeah, I hope I'm not dragging everybody else down, but this is new to me, and I want to make it comprehensible to people who may not realize all this. If I don't, I'm not the only one.
Right, and so this is why I'm glad to invest time in teaching you creationism, very unlike the way you got it in that other place. Okay, because I don't want to advertise that other place. You know, it's bad for the kids. So yeah, bad for women and children both. Sorry, just had to take a jab there. So I hope you're learning something.
Take a sword, Sal.
Yeah. So I hope this has been educational.
It's mind blowing.
You'll be in the position to decide what age group to share this with, because this will probably just blow away like the 4-year-olds, definitely. I mean, I did try to teach some of this to 4-year-olds, and I would, the best I could do. I had my nephew, God bless him. I'd say A is for adenine, T is for thymine, C is for cytosine, G is for guanine, and he'd repeat it, because the kids are just like sponges, and we just do that for half an hour, and then I'd say, okay, now I know my ABC. Next time, won't you sing with me? And so that's how I taught them, just as a song. And so this little 4-year-old we were playing Frisbee out in the yard, and he'd go, A is for adenine, A is for adenine. Okay, A is for adenine if we're talking nucleic acids. If we're talking about amino acids, A is for alanine. But it was just really cute to, this little 4-year-old running around our yard, he's saying, A is for adenine, A is for adenine.
Yeah.
I think I have a video. Yeah, okay.
There was a lady at DAL that was teaching the children things like that. Really big words and high concepts. She was teaching to 4-year-olds in a song. And then what they do is they circle around the next year. They call it spiral education. They teach a little bit about the same subject each year so that they get more an in-depth understanding as they're old enough to get it.
Yeah. I mean, the kids don't care. They're just like, oh, it's so nice, Uncle Sal spending time with us. And so we were just running around playing Frisbee, and he'd be saying, A is for adenine, C is for cytosine, G is for guanine, T is for thymine. And I don't think he got through all 4. I'd have to help him, but it was kind of cute. And so at some level, that's about all I could do for a 4-year-old. And as they grow older, that's what the moms and teachers and parents have to figure out.
Well, I think there's a lot of people in I don't know about your YouTube audience, but my YouTube audience that are maybe college age, but still not understanding this stuff. Depends on what classes you've had.
And there are people who went to Dinosaur Adventureland and never learned this stuff. And I think it's important. So,
yeah.
because
So
now
Sal,
you
can
can
I make
start.
just an overarching generalization? So topoisomerase is microscopic. You can't see it, right?
Correct. We cannot literally see it. And Joe Deweese in the webinar said, we don't know what it looks like, the C-terminal domain. And I'll show you the C-terminal domain here.
It's comprised of strings of also even smaller microscopic molecules, which are also comprised of conjunctions of microscopic atoms, which are also comprised of systems of microscopic subatomic particles.
Correct.
It's infinite smallness.
There is a hierarchy of, we call it, layers of organization. What you just described are layers of organization. And so it's first organized, as you said, subatomic. Then you have atoms. Atoms connect to form molecules such as amino acids. The amino acids connect together to make a protein. So,
and the proteins connect together to make a topoisomerase.
Two topoisomerase polypeptides make a protein. And before we go a little farther, I'm just going to try to insert something here to clarify the diagram. Hang on, just if you'll
those letters aren't called proteins. Those letters are called
amino acids. So let's get back to the diagram. I just had to insert something to help. I had a missing slide and I just inserted it while you were asking. So, all right, let's go back to the layers of organization. So the first layer, the very first layer is obviously the subatomic, and then the atomic level, that's where you get the C's. C is for carbon, O is for oxygen, H is for hydrogen, N is for nitrogen, and then the S here, down here, is sulfur. And then so the carbon connects with oxygen, and then this oxygen ion that has the minus, we call that a functional group. So the atoms connect to make functional groups. The functional groups connect with other atoms and other functional groups to make an entire molecule of an amino acid. And we can call it, roughly speaking, we call it also a monomer, because it's one, mono meaning one part, one. And so when you connect monomers or amino acids, okay, so an amino acid is a monomer. A monomer is not necessarily an amino acid. Just so you'll hear the term monomer used a lot. So when you connect, so the beginning of the topoisomerase is a methionine. So you take a methionine monomer, you connect it to an E, which is a glutamate.
Now,
and then
their little arms, do they actually touch, or do they actually bond, or do they just stand next to each other?
They have a covalent bond between the carboxy group and the amino group. I'm going to mess this up. Someone correct me if I got this wrong.
The M and the E, for example.
Okay,
when they stand side by side to make a string.
I'll tell you how it connects. I'll tell you how it connects. The CO of the COO, the carboxy group here of the methionine connects to the amino group of the glutamate. Let me see if I can find it.
To the head. If you were to think of them as little bodies, how they look like they have arms and heads. So
exactly.
The head
Let me see if I can find a diagram of that.
So that negative symbol next to the O means it has the availability to connect to something.
Yeah, and some of the digging, I may not have used the right terminology. Let me see if I'm looking for a diagram right now to help the viewers out. And this is Cindy, you're now asking organic chemistry questions, and everyone knows
Well, the
organic chemistry.
evolutionists are using DNA as their Trojan horse. And if people don't understand how complex and how specific this is, they're going to be duped.
Oh, yeah. So this is what we call a peptide bond, and I'm going to insert it in our slideshow here so we don't lose it. So just if you give me a moment, I'm actually redoing my slideshow.
Like even if that one minus sign was gone, so that oxygen was just oxygen instead of the oxygen with the minus sign, it wouldn't be able to bond. That's how everything has to be just so. Am I missing
exactly?
That?
And in fact, that's what James Tour was trying to point out. He said, try to make some of this. He said, it's really hard. You can't just throw these chemicals in a soup and expect them to make stuff like this. He said, it's really hard. And even when you get there, there are all sorts of ways that this could bond the wrong way. So let me see if I can get this picture of the peptide bond, which you were asking.
It's kind of,
so I'm gonna,
kind of like a puzzle. It won't fit if it isn't the right shape.
Okay, so let me see if it took, let's see if it took my diagram here. Okay, so going back to our show, back to your question.
Ooh, that's better. There's the O with the minus, a carboxyl group.
Carboxyl group. Okay, I said carboxyl. And then it is the amino group. So we'll connect. Okay, look carefully at the R. All right? The R is the R group, which corresponds, just think of it as the red group, okay?
You mean the pink?
The pink. See, okay, a woman would know the difference between pink and red, okay? Just like a girl got me this orange colored shirt, and I said, I'm not going to wear that orange shirt. She said, no, it's coral.
Salmon.
Yeah, okay. Girls, I mean, physiologically, they have established girls they can perceive color much better than males. The shades of coloring and all sorts of, they can distinguish between them. All the little gradients, you could just, yeah, they process colors in a way that men don't. And so that's why I was just laughing when I said, yeah, it would be a woman that would say that's pink. Okay.
You did
all right.
Pink earlier.
Yeah, I did, but I'm just trying to give a mnemonic here. If they made this red, we call it R for red. All right. So.
Oh, that's why. Okay, I'm sorry.
Okay, so just consider all the pink regions the R group, because pink is sort of reddish. And then, and then you see the COO here and the H3N. Okay, this is where it gets confusing. It is H3N, but it doesn't look like it's spelled COO. You see how it's really the O, it should be OCO. You could also say COO, okay? Because everyone will represent it a little differently. That can be annoying, okay? Are you able to get over that? Here it's spelled OCO.
You also have to realize that you dumped it on its side. So now the head is, the arms, you took away the human lookingness to it, but I can
right. So the R group kind of looks correct here. The R group here is here. Instead of it at the top, they put the COO at the head and the H on the side. So that's what is annoying about some of these chemical diagrams. See, you can see they interchange the COO here and the H. It's flipped. And then you have the R group here. And then to make it worse, in here they make it upside down. Okay, this is why organic chemistry is really hard. Is you have to keep flipping. It was really funny. I remember a physics guy was talking about a physics exam in electromagnetic theory, and you could tell you had the guys in the middle of an exam kind of doing this with their hands. They're trying to conceptualize all the parts. And in an exam, I don't know if the organic professors will let you bring molecular these molecular balls and sticks where you can connect it, because you really have to try to visualize this in 3 dimensions. And the professor will say, describe this bond. So I'm teaching you about all the organic chemistry I know. I actually studied biochemistry.
Well, you were going to what?
I actually studied biochemistry and published in biochemistry journals, but I don't really know organic chemistry. People know that.
You were going to talk about the bonds, how the arm connects to the head.
Right. So they will come together, and there's certain conditions that make that bond possible. I think you need ATP to make the bond.
So that it releases the bond from the other group and hooks on to the neighboring group.
Okay, so what'll happen is when these two come together, it'll make water, H2O, and then it'll connect the two together. It'll fuse them together. And I think that needs ATP.
Oh.
And I'm going to show a video that does this. I could be wrong. Someone in the chemistry section will.
Plus sign. The N3, no, H3. That's why the H2O is released, because 2 of the H's are gone.
Right. And one of the O's is gone. So what'll happen is the O is negative, and the N is positive. And somehow when they come together, all the charges go, and you get H2O coming out, and then you get this bond. It's called a peptide bond. And you need a specialized machinery called a ribosome to do this. And you'll hear us talk about ribosomes. That's another machine.
So wait a minute. Those 2 molecules on the left of the plus sign and the right of the plus sign, if they're just swimming around in water, they will not connect. They need that thing that you just said.
It needs ATP. Because water will prevent, when you have the amino acids in water, water will prevent them from bonding. And that's something James Tour pointed out. He said that's why
the primordial soup doesn't work.
Exactly. And then we were laughing at Dave Farina, kind of the punching bag. It was really brutal because Dave Farina is like a punching bag. He was such an idiot. Sorry, I'm not supposed to say that about people. I'm not supposed to be that was kind of uncivil of me, but Farina did look like a goofball. Goodness. And there was this one graph where he put Dave Farina at the top of Mount Stupid. I'm just like, yeah.
But I'm sure Dave is doing the same thing to him. He called him stupid too. Dave called him clueless.
Well, you know what? James Tour is a famous scientist. Dave Farina is like a nobody by comparison. I mean, Dave Farina is not even a scientist. And he's talking to a chemist, a professional chemist that's possibly one of the top scientists on the planet. I mean, James Tour spoke before. Yeah.
I was really upset. Calling him clueless. I'm like, you must not have watched the lecture. Nobody who watched the lecture could call him clueless.
Farina is a narcissist. And a narcissist will never be wrong. I mean, that's just Farina. No matter, you can't reason with a guy like that. He's an angry, antisocial narcissist,
I
an atheist.
Know that. I did.
Well, that's just my diagnosis, okay?
Yes,
but
Dr. Sal.
Yeah. So going back to this diagram here, this reaction won't happen in water. In fact, if you have this bond, this peptide bond in water, over time, water will get in and separate the two. And that's what James Tour is trying to say.
So what exactly medium are they floating around in where they're creating these peptides? Or
they
whatever
have
you
water,
call
but
this.
then you have machines. You have machines. I'm sorry. You have machines that will stitch them together because it's not a natural reaction in water. You need an outside force. You need an outside mechanism to override what's natural. So
And what's the name of
just
it
like
again?
when you need an outside mechanism.
No, the name of the machine.
Ribosome. A ribosome. And I'll show you videos of that. I need to clear the copyrights on some of this so that I could incorporate it in my videos. When I do that, I could show you this. And so what James Tour is pointing out to Mr. Dave Farina, who's
Yes.
on top of Mount Stupid, he said, well, Dave Farina was right to say, well, then it has to go through a drying cycle because this reaction won't happen in water. So you have to get the amino acids to dry out somewhere, and then they might connect. They won't necessarily. And then they could go back into the water. So you have this cycle of what they call wet-dry cycles. And it's just kind of ridiculous. It's like, okay, so they have to connect. That's the first part. Well, first you have to make them. And that's also another difficulty, making them. And then you have to get them to connect, which is not really natural.
In order for them to connect, they'd have to be close enough that their attraction would attract.
How do they move around if they're dry? So water, this is what they call the paradox of water, the water paradox. Water is essential for life, but it will also prevent life from forming spontaneously. Whichever way you go, water will be a problem because
so will
just
the
like
sun,
what they
and so will oxygen, because oxygen's necessary for life, but it also oxidizes things, rusts them, or whatever.
So you can't live with it. You can't live without it. It's one
kind
of
of like men.
I knew you were going to say.
But you were going to say it if I didn't about women.
Yeah, you were. I was just about to say that. Because I've known some guys who would say it a little worse. They'll say you can't live with them, you can't shoot them, or you can't live without them, you can't shoot them. Although I shouldn't say that. It's terrible. So let's move to, okay. So now we were just describing the connection of methionine to glutamate. So that's just connecting two together.
2 out of what did you say? 5,000?
1,531.
Oh, your paragraph is 1,500. Okay.
31, right?
Yeah.
So you connect
Oh my gosh.
You connect the carboxy terminus to the carboxyl terminus, carboxyl group to the amino group here, and then the glutamate has its own amino group, its own carboxyl group. That carboxyl group will connect to the valine's amino group, and so on and so forth. And so then you get this.
Equally unlikely to occur, even the two, much less the 1,500.
Yes. And that's, now you're appreciating what James Tour was trying to communicate.
Right.
He was saying we can't even get the letters. So that's the first problem. After you get the letters, you could hardly connect two of them in water. You won't. And then he didn't even get to the point. Okay, he only touched on this briefly after 10 hours. He said after you're able to find a way to connect them, you have to spell them right. The other thing is they don't necessarily connect in a string. You could just get this blob. You want them to connect in a string. So just imagine taking a necklace and gluing all the beads together rather than forming them as a string. It's easier to just get them as a connected blob where you just throw them together with some glue and they'll just kind of blob together. You really want them to be in a string. Once it's in a string, then now it has to be able to be folded. So you take the necklace now, so to speak, and now this is what they call protein folding. So now you've learned a lot about the complexity from abiogenesis now even to the topoisomerase.
It's just layer upon layer upon layer of complexity.
Yeah.
I mean, even the folds are obviously done in just the right way. How are you going to do that
so
with
by
a
the
necklace?
way, you've just blasted through about 3 years of college right here, the essentials. And this is what's important that I try to say is that we can actually teach some of this within less than 10 hours, the essentials of understanding.
Yeah.
You just comprehended, you can appreciate the complexity. And so this is why I've been encouraging you to study the molecular level arguments. You will not, everything that I said, someone like James Tour will back you up. And he'll say the other side, like Dave Farina, it's on the top of Mount Stupid. Because you could hold this, you could hold your own with this. And this is, when I saw your video, Dapper Dino's video on your Science Center tour, he probably wouldn't have
too many holes.
That same complaint if he saw this, because this is out of textbooks. Okay, everything I've shown is just right out of the textbook. This was from my Leniger biochemistry textbook. Not going to complain about that. This is from an organic chemistry textbook. That's real. This is from just, this could fit in any science textbook on biochemistry or protein biology. And this is from a database that's inside the National Institutes of Health. There's nothing here that we've gone so far that's controversial, and that's what I was trying to say. I know you have your love for Inca stones, but
Ica, not Inca. Not Inca. It's a different civilization. The Ica are before the Inca.
The what?
The Ica came before the Inca.
Oh, sorry.
I believe.
Okay, but the point is, everything I've shown you so far is not. They're not going to question the authenticity of what I just presented.
I see.
So you have a good starting point. You can then argue why this is not reasonable to evolve. At least you can agree on what the facts are. And you're in a stronger position now to make your case. And even as you're watching this, I could tell you're kind of blown away by the complexity in each of the steps. So, and by the way, remember, topoisomerase is only one protein in the human proteome of 20,000 at least. Okay.
Each have a specific spelling paragraph that only works. It's a signature spelling paragraph.
Right. Now I'm going to tell you something.
Fingerprint.
Right. I'll tell you something even more amazing. This topoisomerase here, you see the red and the blue? Each of them are identical copies of this. I use the term protein. Polypeptide is probably a little bit, strictly speaking, a little bit better term. I'll call it a polypeptide. So you need one polypeptide copy. The gene will make one copy, and that's the red copy. It'll make an identical copy. That is the blue copy.
That paragraph is only the red?
It's the red and the blue. You make two copies of the paragraph. So
that's
the
what
ribosome.
I say. So that picture of the red and blue would need two paragraphs.
Well, actually, you need one because it's a copy. You have a copy machine process in the cell.
But in terms of quantity.
Right. So you can make. Why can we do that? We have the DNA. DNA is a blueprint, and then the DNA makes what they call RNA. The RNA then can make with the ribosome make the protein or polypeptide. So it's just like hitting the print button several times. Oh, Nestling's here. Oh my goodness. Nestlig is very smart. He's an evolutionist, but he's very, very smart. He'll correct me when I'm wrong. So you're able to make copies of this. So to make a functioning topoisomerase, you make 2 copies. And when you make the 2 copies, and Nestlig pointed out, you call it a dimer because it has 2, di for 2, mer for meaning it's 2 pieces together. So that's how you make this. You know, if you have a, let me show this. If you have
so the copy machine is not just, it's actually creating matter then, because it's making another one?
No, it's not creating matter. It's just like a copy machine that takes ink and paper and makes a document based on a blueprint. Or let's say when you hit the print button, it's going to take the paper.
Yeah, but the
and
protein
ink.
is an actual thing.
Yeah, the protein is made of amino acids that are floating around in the cell. And you know, when I get the copyright. Okay.
When you make a copy of it, you're increasing space and mass, right? You're making something else. You're making a duplicate.
No. Okay, so when you hit the print button on your computer, it's connected to a printer. You're not making mass. You are rearranging the matter. So the ink is in the ink cartridge. The paper is in the paper feeder. When you hit the print button, it's going to join the ink to the paper, and you have a document.
Okay, so it's going to use its surrounding materials and arrange them
to make a document.
When it's making a copy, it's somehow reaching out into its environment and getting all the letters that it needs.
Right. Where are the letters
those
stored?
Letters into a paragraph.
The letters, the blueprint for this, the way it makes this necklace, so to speak, the sequence information is in the DNA. So the DNA is like, you might say it's like the file inside the computer. And when you want to print the file out, you press the print button, and then out pops your document. It's an analogous process where the file is in the DNA, which is the blueprint. And every time you press the print button, it's going to make a every time you press the print button on the file for topoisomerase, it's going to make the topoisomerase polypeptide. Does that sort of make sense now?
But it's a thing.
It is a thing, just like the paper in your printer. Yeah.
Okay, so it duplicates itself in order to replicate the cell. Is that correct?
Not quite. So let's back up a little bit. You have kind of the file. You've printed out files before, right, on your printer?
Yeah.
Okay, so you have to load ink into your printer, and you have to load paper.
Yeah, that's what I'm saying. So it's taking the letters from its environment. It's taking the ink from the ink cartridge and putting it on the paper.
Right, according to the instructions of the go on.
So it's taking the M or the whatever the letters are. It's taking those from the ink cartridge, which would be somewhere in the cell environment, and putting them into a string.
Correct. And the way it figures out which to put first and the next second and third, it's controlled by the ribosome, which gets instructions from the DNA. This is a complicated system to be able to keep. So you could take one file on your computer and you can make a bazillion copies as long as you have enough paper to keep. You have to keep feeding your printer paper and ink. And the cell is very similar. For it to synthesize these polypeptides and proteins, it has to be fed and nourished so that it has the supply to keep manufacturing. A lot of the energy of the cell is actually, a lot of the energy of the cells is expended in constructing proteins. This is a very, very complicated process. It's unbelievable.
So the raw materials come from us eating.
Right.
What we eat goes into the cell, which is like the ink cartridge.
Ink cartridge and paper, so to speak.
Uh-huh. Wow. Okay.
And of course, the analogy is the metaphor will break down at some point. Now, I have a pair of scissors here. It's not really symmetric, but you actually have some scissors that the parts are absolutely identical. Each scissor
Yes.
can be identical, and then you just connect them together. And that's what the topoisomerase, you need two symmetrical parts. And it does look like a pair of scissors, doesn't it? And you saw how it worked. And the cutting part is actually somewhere like right in here.
Yeah.
Okay. So it's a close approximation to a pair of scissors, but it does more. So when
scissors
you have
with a brain,
it's more like a robot. It just seems to know how to do what it does. So, but the way it connects, like unlike my scissors here, see it's connected by the screw in the middle.
Uh-huh.
The topoisomerase connects
like more like a hinge.
No, I'll show you the connection points. Let me show it to you. There are all sorts of points where it connects right down the center. And these are, this is part of my research. For a paper I'm doing, there's this software tool called PISA, and it can estimate the connection points. And you can see all the letters that are involved in connecting the two pieces together. Now, I won't vouch for this. The software tool could be off, but there are about 60 points where it has to connect. And it has to be about the right strength. Because when we saw how this thing was moving, it was like a hinge. So some of this will actually separate, and so, depending on the state, depending on the on what phase of the process of cutting and doing its thing, some of this will connect and deconnect. But it has to be pretty precise. There are subtle charge differences between some of these. Some of them will tend to have like slightly negative and slightly positive, and that enables the connection. And there are all these other sorts of bonds like van der Waals interactions. I mean, I can't even. This has to be precise within angstroms, which is like the size of the radius of an atom. It is so precise how close they have to be so that they can connect. It's unbelievable.
And then something probably has to happen that will break that connection.
Exactly. So I want to point out, this is showing how the spelling, another thing is spelling helps dictate the shape. So if you spell this different, you won't get these shapes. So how does it know that I need to spell it a certain way so that it will connect this way? And in a way that it will enable it to do all the things that you saw in that 1 hour lecture. Just amazing. So this is the spelling of the topoisomerase, and I just colored the connection points. But then Dr. Deweese was talking about the way you could arrange, you could identify. And so this is from a paper I'm working on. He's talking about all the stuff. So you heard about the ATP, that powers, how does it powered? This part takes the fuel. So this is like the engine right here, the ATPase domain. And you heard him talking about how it connects to metal. And there's this little section here I called the Dairy Queen. See, DQ, DQ. That was his PhD dissertation, these two letters. He had to figure out what DQ, DQ does. And he was talking about, I think these have like negative charges. So D is aspartic acid, Q is glutamine, I think. I'm embarrassed now. I have to verify that.
I was
Q,
fascinated when he was talking
Q's
about
glutamine.
magnesium.
Yeah, magnesium will
how
connect right here. Right here.
magnesium is a metal, and I was Googling it. It's a fascinating metal that naturally goes into like the shape of a feather. So what you're saying is this requires like one atom of metal to be part of that
Right,
molecule.
complex. So the ATP, the gasoline, so to speak, is here. That's right here. And the ATP will, I think it'll be either here or here specifically where the gas molecule goes. The cutting edge is, to enable it to cut, is right there with the magnesium. And so when I'm with Dr. Deweese, he'll be showing the diagrams. He'll say, okay, you'll see like that DQ right here, and he'll identify. So I would think the DQ is kind of like in the center, somewhere around here. That's magnesium.
The cutting surface.
The cutting surface. The ATPs are here, and he's talking about it's ATP here and it becomes ADP in one part of the cutting phase, and then when it's doing the reconnecting, it's the other part here that gets involved and gets converted and consumed. It goes from ATP to ADP. So I'm just like, I talked to biochemists. I said, if you didn't know how to build one of these, we know how to build one now because we have the spelling. Could you do it from scratch if I didn't tell you the spelling? No one has said they would know how to build something this complex. And it's
ridiculous.
Yeah. See, now you know I'm glad you're learning more about the molecular stuff. And you only need, Darwin said you only needed to find one system and this theory would collapse. That's why I said just topoisomerase will do the trick. And then once you learn, now that you're learning about proteins and polypeptides and all this other stuff, you'll be versant in knowing how to do other proteins. So I said these are the connection points here. So this is what's interesting. Remember, you have the connection points, but then somewhere here, and you have the DQ here, and you have the ATP binding somewhere here. That's why you need these different color schemes. And so like Joe Deweese is talking about where DNA also contacts, you have these DNA interaction points, like here and here. I mean, this is so hard. You can imagine how difficult the experiments had to be to identify specifically what amino acid touches the DNA in the cutting process and processing.
Well, and it's only one letter.
Yeah.
One letter right in the middle of the paragraph, not even touching the rest of them all.
Right. So this is a tyrosine here, this Y. The S is a serine. And then you have this,
so
this
take
one
that
here.
out and it doesn't function.
It may. I'm pretty sure that when Joe Deweese said that he could knock out one amino acid and it'll break, he's talking about the DQs here. I think if you knock those out, it's topoisomerase is gone. There are other parts of this you can mess around with. You heard the term C-terminal domain or carboxy terminal domain. So this region here, you could sacrifice a little bit.
Oh, that's right. That's where he was talking about the alpha and the beta. One of them could work out of it and one of them could not.
And neither would work very well without the other partner. Okay.
Yeah.
So this is the C-terminal domain here. See, this is, now you made me really glad. I got to tell you something. I kept thinking, is it a waste of time for me to put diagrams like that together? Because this took me a day to put together. I had to go through all these databases to identify the parts, and then I had to color it. I mean, I had to dig through all these papers. This was just painful. And there I am, I'm just coloring all the little parts. I'm the one who built this diagram here. See, now you're making me feel like, huh?
Nerd torture?
Nerd torture. You got it. So I didn't do the experimental work. Someone else did, but I had to comb through all their experimental papers that would identify all the parts. Thankfully, I had Joe Deweese.
Yeah. And you flip it up on on the PowerPoint for less than 30 seconds, and very few of your watchers know what the heck it is, and biff, it's over. So yay! I'm glad I got to make your whole day's thing like come alive a little bit.
Yeah, because, you know, people look at this, it's like, okay, it's just up there
What
for 30
is it?
seconds. Like, yeah, it's a day of work, but now you see all the parts. I actually don't know what the transducer domain, the purple, does. And then you heard Joe Deweese, they're talking about their places the topoisomerase wants to go. And he said there are two nuclear localization signals. And those two are right here. Now it's only one red blob, but you know, there's one part here and then another part here, something like that. So it's actually two, but I represented it as one because it's kind of in the same region. And it's called the Bipartite nuclear localization signal. In some publications, it will make it either one or two. So I just for the ease, for the sake of easy being, you know, just for the sake of simplicity, I identified. So this is the address. This is the postal address right here to go in the nucleus. He also said there's an export signal that will actually send it outside of the nucleus. How it knows when to use this postal address versus this one. So it has something that tells it when to go outside, when to go inside the nucleus.
Those
and how
you
that's
were referring
deployed.
to as like passwords in your other video with the other cartoon?
Yep.
So you're saying, how does it know which password to present?
Right. Exactly.
Another one of the big questions.
Oh yeah, and he said that you know, I think he would. I know him enough that he would. I think he said we've interacted because we worked together. He said, I don't think we really know how it's deployed. You know, how it knows when to use one password versus another. Because just like when you're going in and out of places, you might have passwords that you enter out. This is a very regulated process. And let's see, he used the word top rim domain. This is the topoisomerase primase domain. And honestly, I don't quite know what that does. So you could see that we began all the way back here with the atoms, carbon, oxygen, hydrogen, nitrogen, sulfur. They make amino acids. The amino acids connect together with peptide bonds. Then that makes the polypeptide, but the polypeptide is itself made of critical, you know, you can identify the parts organizationally. These are the connection locations. The reason they're different colors is it's different experiments that were involved in identifying the connection points. I mean, this was brutal work for the experimentalists. So you needed one set of experiments to get the yellow points, another for the green points, another for the purple points.
It's kind of like us going against the atheists. We can only do one little point in a whole big, huge avalanche of data coming against us.
Yeah. And then you had the other researchers, poor researchers, that are identifying the other parts of how they work. So, and you can also see that they're overlapping. You know, you could see some of the connection points would be in the colored regions here. This is really, to engineer this is just insane. And I said Joe Deweese had worked 20 years of his professional life on this, but there are hundreds of researchers. This has been their whole life. This 1,531 amino acid stretch has been their whole life because it is so complicated to figure out. And so the topoisomerase alpha is
1,531. So to figure out one element and what it does, then you have 1,530 to go. And if you figure out two, then you got 1,529 to go. Is that what I'm getting?
Well, it's not just that, because you can identify them, like in this case, sometimes you just have one amino acid, but then sometimes you have whole groups of them. So it's just for one, and then also working it out for how it works as a whole, and they're still working on it. And I haven't even, there's even another layer of this that's, okay, because I promised I'd talk about my research, and we're almost at the 2 hour mark. And we've already been studying topoisomerase for 1 hour with Joe Deweese, and now 2 hours with me. So you heard him talking about alpha helices and beta strands. So the alpha helices and beta strands, let me show you the picture. So when you see these curly things, those are alpha helices. And when you see kind of the line things, those are the beta strands. So we can identify it in this diagram here. And so when they turn, see if I can get the picture here. We can identify the points that they turn, like right there. So you can see it's going one way, and then it turns around and goes the other way. So we're able to identify the turning points, where the strands are, and where those curly things are. So wherever you see this yellow, that's where it's curling. You can see the strands by the green, and in the regions where it's turning by the red.
In other words, the folding.
The folding, yeah. Well, the proper term is the secondary structure, but it leads to the fold. Right. So collectively, you take all these parts, and where they turn, you get the fold. Now, he said the C-terminal domain over here, you could see it doesn't have any coloring. He said that's why we don't know the shape. So the way that they're able to figure out the shape is they have to use these experiments, because we don't have microscopes that can actually see this. Very hard. And he said, to this day, we don't know how this is shaped, this part here. He said, for all we know, it may just kind of flop around and change shape. And I think both, I could speak for him, I think, that the shape can actually, it depends on kind of chemical actions on this region. It'll affect the shape.
Yeah, he said the shape morphed.
Right, and I'll describe that in the next. So let me just pause here. What do you think so far? I haven't even touched on, I have another layer to talk about, but we'll just take a break.
Yeah. I guess I'm just blown away about, so first you have to get them to bond in the right order. Oh, I guess what I was thinking, you know how they talk about junk DNA because they don't know what it does. Well, what if it just tells it where to fold and it doesn't do anything? It's just an indicator.
The problem of how these things, yeah. Okay, so the DNA, what we call the coding DNA, is basically the file that gives you, you know, when you're printing out a file. So just imagine the DNA is the file in your computer. And it has the recipe for the protein. So the DNA will then get translated. It gives the letters, the sequence for the protein. The junk DNA, I don't know for a fact that it does much to actually fold this. We have a chaperoning process that will, folding mechanisms that will get the necklace, so to speak, to fold the right way. Sometimes it will spontaneously fold, but there's way more to this. So Ben Rex said evidence and reasons, and then there are people paying for other people spending their entire life on this research. One reason that people are willing to donate money to this research is this is an important target for cancer chemotherapies.
Yeah.
So that's why a lot of money can be poured into this, because it's trying to save lives. And in the process, we learn about God's creation. We learn, I mean, when you just see this complexity, it's like, yeah, they asked Joe Deweese, do you know people that try to describe the evolution? And he was polite. He said, yeah, there's some people that try to do this. And I wanted to say, and they're not doing a very good job of explaining it with evolution or abiogenesis. And for the reasons like, okay, for abiogenesis, James Tour already outlined why it's so difficult for abiogenesis. He was just pouncing on Farina, Farina on the top of Mount Stupid. He was starting from the basic chemistry. So this has to be overcome to make them themselves.
Well, and
then
the
the bonding problem. I'm sorry.
That the sad thing is, I don't know how many people will just see Dave's thumbnail and not even plow through the science and just assume from the thumbnail that James Tour is clueless, because that's what Dave's thumbnail says.
Yep. Now, I found something really interesting on the internet by a very good psychologist and a investigative reporter. They said they're talking about girls and anorexia and also kind of gender dysphoria issues. And she's pointing out, how does this spread? How do harmful behaviors spread? Certain women definitely have a certain proclivity or tendency towards certain harmful behaviors. But anorexia, she said, if you're treating anorexics in a hospital, you have to make sure they're kind of separated. They found out if they're in the same ward, you have to be very careful because they'll start to reinforce each other's bad behaviors. Meaning, we're trying to treat the anorexics to say you're not fat, you're actually pretty thin. But if they're in the company of other anorexics, it'll reinforce their viewpoint that they're fat and they need to stop eating or do something. Mm-hmm. So the point is, when you get people that have a certain tendency and they're able to connect, they're going to reinforce each other's bad behaviors. And the internet has really facilitated that. Were harmful, and she specifically said her name was Abigail Schreier. And I had a clip on this channel a few days ago where it was her and Jordan Peterson talking about this. They said the way these women will experience friendships and bonding, they'll even suspend reality in order to maintain the bond. So you could see how this would work in anorexia for the girls. They're going to suspend reality that they're actually thin, and tell each other we're fat because the bonding is so important. This will happen also for guys for other things. But for bulimia and anorexia, there's these pro-bulimia, pro-anorexia websites, and it's just destroying girls. Dave Farina is kind of in the pro-abiogenesis crowd, and they will suspend reality. So it's not just a thumbnail, it's a culture of denialism. And the internet is an ideal place for these people to congregate and just kind of deny reality that James Tour was teaching. And we're only talking about organic chemistry.
Well, no, I can think of some others.
Yeah, okay. Absolutely, yeah. We could think of other things.
But I see what you're saying. You're saying that they reinforce each other mindlessly, pretty much. And they would say the same thing about us.
It's the herd mentality. Yeah, they would say, and to some extent, sadly, it's true. I mean, I see it in the church groups where the pastor, need I say Dinosaur Adventureland? We see people congregating and they've been told the truth and they're like, oh, he's the Lord's anointed. Doesn't matter how much criticism, it's so obvious, the abuse of, the questionable treatment of children.
A lot of us hadn't seen it all, Sal. I hadn't seen all this.
Okay. Yeah.
I had not seen all this. In fact, I did an interview the other day with a guy who had been following him since he was in jail, and he said that he has evidence and proof that he lied to the judge in the courtroom because the courtroom transcripts are publicly available. And I'm just like, why didn't I see that?
Yeah.
Anyway,
okay, so
he's about to publish it. I'm really glad about that.
Yeah. But it is fair to say there is definitely tribal mentality and the suspense of reality. that's why I like science. You can't run away from the facts. And this battle between Dave Farina and James Tour, they're arguing over text. A lot of it's just arguing over textbook chemistry. And it was really hilarious to just see James Tour put Dave Farina on the top of Mount Stupid. It's like, yeah, you're putting him in his place. But this is why I'm glad to talk about it. We could go back and forth about who's who, but I just walked you through the chemistry. And I'm relatively confident that, say hypothetically, you went on to study general chemistry, then organic chemistry, and then the biochemistry, that everything James Tour said would only be reinforced, because that's the way it worked for me. And the other thing is, you could see the difficulty in terms of the probability. This is why there are now more peer-reviewed, there are more and more peer-reviewed scientific papers that are saying we need multiverses to solve the problem, because you could see the difficulty.
Yeah.
Prior to the multiverse solution, it was the space aliens. Okay, so.
And the punctuated equilibrium.
Well, that's not chemistry, but anyway, you keep seeing.
No, but it's
this
an excuse because their original hypothesis didn't work. A lame excuse because their original hypothesis didn't work.
Oh, wow. Yeah. So really, this whole debate, creation, evolution, on both sides, especially when we have characters like Kent Hovind, it points out people's ability and willingness to sometimes suspend reality in their judgment. So when we're talking about characters, you know, to this day, people call him the Lord's Anointed. And I think, like you, I want to say, when I hear that, I want to vomit, you know, and it happens. So I remember when I was walking through the chemistry with a biology student at James Madison, and I was explaining some of this stuff. And, you know, she had organic chemistry and biochemistry. At that time, I didn't, but I knew some of the James Tour's arguments. She was in disbelief because she can't believe that her professors would believe one thing and teach another. And
this is exactly like the cognitive dissonance that I lived with at DAL, only on somebody else's shoe.
Yeah. Yeah, it happens. And so I've had to work through that where, you know, I'm having to fight my own tendencies to agree with other people and still be a member of the tribe. And you, speaking from experience, know how hard it is and how much you have to go through before you say, no, what I'm hearing about this or that isn't reality. I'm just being pressured to believe it. And for a long time, I believed it. Until you get a fact that is so incontrovertible, either through reasoning or your personal experience or both, then you're able to reject and say, no, what everyone believes is false. What everyone in the tribe believes is false, either about this human being or a claim, a scientific claim, etc. And it takes a lot to overturn it.
To be kicked out of the tribe is a very scary thing. And that comment that you just put up on the screen, I think his name was Ben Max, how the woman with anorexia, if she put on a different pair of pants, she would be shocked and not understand why they didn't fit. It's like reality versus the brain. Our struggle to keep a false reality. It is just bizarre.
Yeah. And there's some things that are more innocent than that, especially being a pilot. Your senses can fool you when you're flying because we're not really designed like birds to fly.
Right.
And that's why John F. Kennedy Jr., he flew his airplane upside down and flew it right into the water and killed him and his wife and niece. He was a little bit too cocky, unfortunately. but his body was playing tricks on him. His instruments were telling him the truth, but he was relying on his instincts. And the heart can be deceitful. And so a lot of things are the way we perceive reality. And we have to try to synthesize all of our sensory inputs to say, what do we trust and what do we not? And you really, having been in aviation, that is so key, is you have to trust your instruments. Your eyes will tell you the truth if you use your eyes to look at the instruments. But your eyes will deceive you. If you look out of the cockpit and it's a very dark night, you can't see the horizon, you'll see all sorts of things. It's really scary if your airplane is really being rocked around bad. Like I had the case of where I had a pilot friend where I worked. He was in a small airplane, a big jetliner passed, and he flew right into the wake. He got flipped upside down. He didn't know it. Okay, because normally you would know when you're upside down, but when you're moving so radically, you don't know up from down, because you're being shaken around so badly, you don't know that you're upside down. He said he realized he was upside down when the key was, because the key
was hanging up.
Okay, now how can that happen? Well, it was a pitch black night. He couldn't see the horizon. And he should have looked at his attitude indicator to know he had a bad attitude. And there's this cute cartoon. It says bad attitude. It actually shows what it looks like. Let's see if I could bring that up. It's kind of cute. But what told him he had a bad attitude, so to speak, was his keychain. So let me see if I can show this. Bad attitude.
We had a lesson in the science center with the instrument versus the instinct, just exactly like you're saying.
So there are these shirts that they sell in pilot shops. And I love it because it's really cute. Let's see if I could show it here.
The real world is
this is a bad attitude here. Okay, so the blue represents the sky, that orange salmon coral color is the ground, and this is the center is the nose of your airplane. He's almost upside down, heading into the ground, and this device is called the attitude indicator. And if you see that, you got a bad attitude. In JFK,
is that a play on words for altitude, or is that really a gauge called attitude?
It's called an attitude indicator.
For real, it's not a play on words for altitude.
No, it's not a play. So when they say attitude is your orient, so attitude means in the aerospace world, attitude means your orientation.
Towards death.
So you could be at a high altitude, but you're
towards death or life.
Right. It's a beautiful. That's why I love this picture. It's literal and also metaphorical. A bad attitude, you could be headed for death, destruction. And I don't, so attitude is your orientation of the airplane, where you're tilted, whether you're upside down, etc. So going back to the story of the guy, the first thing you notice is his key was going the wrong way. He could have looked at the attitude indicator here and known the same thing, but that's how he knew he was actually upside down. And he was actually flying upside down for a little bit. Didn't know it. How could that be? Because, if you're hanging upside down, you have your seat belt, you ought to be able to feel it, but you're strapped in sometimes tightly enough, you might not notice it. And I've flown upside down. It's like, yeah, sometimes you could be rocked around enough. You actually, if I closed my eyes, I wouldn't know that I was upside down if I didn't see the horizon. Now, usually when I've flown upside down, it's during the daytime, because then it's really easy to see if you have a bad attitude. But if you're in the clouds and it's a dark night, like what happened to JFK, you may not know you have a bad attitude. What did he do? he forgot to look at his attitude indicator. As far as we could tell, his instruments were working. The accident investigators examined the instruments. They survived. The instruments survived the crash. And they said there's no indication that he had an instrument failure. And so he flew upside down, went right into the sea, killed himself, his lovely wife, and his niece. Tragedy.
Failure to receive input from outside sources.
Yeah. No, JFK was not instrument flight rules qualified. He was not IFR qualified. And his flight instructor said, I want to fly with you. Let me help you with this flight because you're not IFR qualified. This is going to be a difficult flight. And JFK, being cocky, declined the offer. He should have had that extra pilot there to help him. He would have been alive today if he had a bad attitude on a number of levels, okay?
Like I said, failure to receive outside input.
Failure to receive outside input. Okay, so
time to wrap
we're talking
up.
about, how did we get to this topic? We're talking about Dave Farina and tribalism, how it can lead us astray in determining reality. So I've been in situations where Christianity was sometimes just pushed on people, and again, they were using tribalism to instill it. I nearly left the faith because I realized a lot of this is just bullying and tribalism, and I didn't like that. And I tried to rebuild my faith. A lot of people prayed for me, so God rebuilt my faith, but he used intelligent design, the study of science, to rebuild it, and we're looking at the topoisomerase. So we were talking, how we got to this topic is how does Dave Farina ignore all this? It's tribalism, self-delusion, and narcissism. That's how that community maintains it.
Because you were saying how James Tour just put him in the dust, and yet he calls James Tour clueless when nobody could call James Tour clueless. And I saw some people saying John Sanford was full
clueless.
Of errors also.
Yeah,
I'm like, oh my gosh. It was somebody we both know too, Sal.
Yeah. So now I'll give you the other layer of complexity because I have to wrap this up. I'm running out of gas now. So we have these letters have significance. Okay, so we saw the letters could be used here. You can identify the connection points to do this, what they call dimerization, which means basically connecting the parts of two, connecting two parts. We call that dimerization. And then all the major components here, like this one is kind of the engine, so to speak. You have the cutting edge right here that connects to magnesium. You have the addresses here, the localization signals, the nuclear. I'm sorry?
Passwords.
Export, and then all these other parts that I'm not that familiar with. And then we have kind of the organization in terms of the little ribbon diagrams here. You see the beta sheets here, the turning points, and then the alpha helices with the ribbons. And that's described by this coloring scheme. And then next is the post-translational modifications. This is a big deal. So the way to describe a post-translational modification is with Christmas trees. So you have the basic topoisomerase, that would be the bare bones, but then you can put ornaments. So what'll happen is you can actually put, in addition to the amino acids, each of the amino acids could have a little ornament hung on it. Does that make sense? That ornament is an additional chemical. So what sort of chemicals can we put? See if I can read this right. On the purple parts, you can put phosphorus. So you can put a phosphorus ornament right here. Phosphorus, where it's purple. The K's, let me see what the K's can be.
Potassium?
K is for lysine. This is where it gets really confusing. So K, so the lysines can get, they're usually acetylations. I should have put a different color. I should have used the green color. Actually, this is just good that I did this, because the green color, I used red. I should have used green or a different color. Those are acetylations. So there's a molecule called acetyl. It's an acetylation. Yeah, similar, I would presume. So you could put all these Christmas ornaments, so to speak, acetyls on the K's. K stands for lysine. S is for serine. And sometimes you can add a methyl group, methylations. You have methylations, acetylations, and then things called summolations. And there's another way I could represent this with a diagram like this. So the human topoisomerase spelling, this is just a section of it. And you can see the chemical on the C-terminal domain that we talked about today. You can get all these chemical modifications. They end up being like bits and bytes, little bits, little flags, bits.
Or like
so
charms on the necklace.
I'm sorry, like what?
Charms on the necklace.
Oh yeah, I never thought of that. I like that. Charms on a necklace or Christmas ornaments on a Christmas tree.
Because I was thinking about how they hold hands. It looks like a line of soldiers, but then you can hang something on their feet or something.
Yeah, exactly. So these are chemical modifications. We call them post-translational modifications. It changes the shape. So when when Joe Deweese was talking about how the C-terminal domain can morph, this is a section from the C-terminal domain. And depending on what ornaments or charms you put, it'll change the shape. And it's also very informative. So that's how it kind of may know what password to use, whether to use the localization password that puts the topoisomerase into the nucleus, or the export sequence that moves it outside of the nucleus to do something else in the cytoplasm. And so for lack of a better word, by modulating the configurations here, you can change the way the topoisomerase behaves. It's now like a Swiss pocket knife. And Ben Rex here is from Switzerland, so he might appreciate that. Did they call the Swiss pocket knife a Swiss pocket knife in Switzerland? I'm just really curious. Ben Rex, so now the topoisomerase can actually do multiple functions by changing the post-translational modifications. And I will add, there's even more to this. They have what they call co-translational modifications. And then even before that, they have alternative splicing of the gene. This is just nuts. I mean, there's so much complexity in the human. And when Joe was saying that on those experiments on the mice where you take the alpha and beta form and it'll compromise the mouse.
Yeah.
And how they're switching, they're doing genetic engineering just to tinker to see what would happen. You can take the human topoisomerase alpha, put it in a yeast, the yeast will live. You can take the human topoisomerase beta, put it in the yeast, and it will live. The reverse is not true because of that mouse experiment that showed it. Now, thank God we won't do this on humans. Some people have enough ethics not to do this widespread on humans. But basically, the mouse will die. one way or the other. You either kill the cell or the mouse you'll kill the individual cells, or if the mouse lives, he'll have neurodegenerative diseases and he'll die.
Yeah.
So he'll be killed either way. So what that means is you can't take a yeast topoisomerase and put it in a rat or a human, but you can go the other way. So what does that mean? The human is way more complex than the yeast. Oh, and Ben Rex answered my question. So the Swiss pocket knife is called a Sackmesser. Sack for pocket and messer for knife. Sackmesser.
Pocket knife.
So he calls it, yeah, they don't call it a Swiss pocket knife. They just call it a pocket knife. So thank you very much. There's the authority right there from Switzerland.
Sack knife.
Sackmesser, a pocket knife. So the complexity of this just is, the more we studied it, and we studied it from the very bare bones, right from the atomic level, the organization is very difficult for the abiogenesis, and then on top of that, the sequences, and also the peptide bonds to get the sequences right, and then all the stuff that has to do to fold and then cut and do all these things. And now we get to the other levels of isoforms,
Adding
which
ornaments.
we absolutely don't have time. There are ways we can get the topoisomerase to be a little different because the gene can be, it's like you have these files and you can just print out parts of the file. That's alternative splicing. And then you have possible co-translational modification, which even I'm not that familiar, where they can change the protein kind of on the fly. I don't know if topoisomerase has co-translational modifications. We do have isoforms that are done.
Be kind of like how there are different kinds of cells? There's a tissue cell, a blood cell, a bone cell. So these are different topoisomerases for different purposes?
Yes, because they're different, what they call alternative splicing, and I didn't even have one on that. But short answer, yes. And when you went through our flip-on nerd torture session, and I started talking about, isoforms, yep, that's it. And then on top of all these alternative splices, possible co-translational modifications, we had the post-translational modifications, which I showed here, which is, like you said, the charms on the necklace or the ornaments on a Christmas tree. And there are all sorts of post-translational modifications. You had the phosphorylation, glycolization, ubiquination, simulation, disulfide bonds. Oh yeah, I had
so
that. So
those all must be for a different specific purpose.
Yes. So where's the disulfide bonds here? The disulfide bonds okay, so you see here the disulfide bonds with the, oh yeah, the lysines have disulfide bonds. No, it should be the cysteines. I missed the disulfide bonds. I didn't even put them in my diagram. Dadgummit. I need to correct that. So there's going to be disulfide bonds in here. I just didn't identify them. I mean, this is just, I didn't even put those all in my diagram here. I don't know that we have any lipidation or glycolization to our knowledge, or hydroxylation, but we have methylation, we have acetylation, we have disulfide bonds, we do have simulation, and I need to put that in my diagram, the simulation points, the ubiquitination, we have that, and the phosphorylation. And by the way, you heard the word ribosome. Here's the word ribosome. So you have the DNA that makes mRNA. It goes to the ribosome, and you make the protein. This is just unbelievably complex, and we've crammed it in about 3 hours.
Yeah, if your mind's not blown yet, right? I mean, I just felt like, wait, you can't get any more complicated.
All animal and experience. Oh boy. It abolished imports of stuff with the animal and human experiments as well. I think most people know that even the coronavax is done with human experiments. Whoa. Okay, just a little bit. people wonder why we're made similar. Why did God make creatures like chimpanzees that look similar to us, and mice have some similarity? I think it's so we could understand our human biology. God gave us the gift of these creatures, these innocent creatures that we sacrifice so we could understand our own biology and cure ourselves. Oh, and Mark O'Connell. Yeah. So I hope you've been blessed, Cindy. It was great spending an afternoon to teach you a different brand of creationism than what you learned at Dinosaur Adventureland. And I hope you've not regretted what you've learned.
Oh, it's just always amazing to study the Lord's creation. I mean, it's mind-boggling how small it goes and how big it goes. To me, it just screams God.
Amen.
A mighty
God.
Yes.
As we close down, I'd like to thank all the people in the side chat. Ben Rex, it's been great hanging out with you finally during a time in your time zone out there in Switzerland. And Mark O'Connell, I'd like to thank the people that showed up earlier, like Schnoil. His mother's going through some ailments, and we're praying for her. You're in our thoughts and prayers. And I'd like to thank Logical, Plausible, Probable for dropping in briefly. And everyone that joined the Zoom call, Patrick Alexander and Harry Stark was here. Even Nestle dropped in. Lots of people have dropped in and out. I'd like to thank all the visitors, and especially Cindy Lincoln, who asked really good questions. It gave me a chance to practice practice my presentation. And Ben Rex says, I've been blessed. Nice thing to do on the side while also trying to do my taxes. But I have to watch the first half again as I didn't pay it. Well, and I do have to say something. The rest of the world, on the other side of the world, there could be war breaking out between Russia and Ukraine. There are all sorts of problems we all individually and challenges we're dealing with. And if it's not us ourselves, we could have a loved one or friend going through challenges. But God said, consider the lilies of the field, how they neither toil nor spin, how they grow. They neither toil nor spin. Yet I tell you, Solomon in all his glory was not arrayed as one of these. Each of those lilies of the valley have topoisomerase in them, as well as humans. So let us consider the lilies of the valley that have topoisomerases in them and numerous other molecular machines that we can't even fathom. Today we have just studied one protein. The plants have thousands of them. And they're
working cooperatively.
And proteins are only one class of biomolecules. We have lipids, we have glycans, we have carbohydrates, and then we have the nucleic acids. We've only scratched the surface. And Jesus said, when he was encouraging people not to become anxious, he said, consider the lilies of the field, how they grow. They neither toil nor spin. Yet I tell you, Solomon in all his glory was not arrayed as one of these. So the command from the Lord himself was to study the way that God made nature. And you'll be comforted that we have a creator that is in control, that is all wise. And, this is tough medicine for me because I'm going through some challenges with my business. And I shouldn't bellyache because there are people that are going through way more than I am. There are brothers and sisters there on the Ukraine border. It could be Christians on the Russian side and on the Ukraine side, and they're caught in the middle of a war. The Christians all over. And so I'd say the contribution of what we've done today is to do is what Jesus said, is to consider these things. We have considered a part of the lily of the valley because topoisomerase is part of all life, as we learned today. And I hope you've been blessed and encouraged, the viewers. Cindy, if you have any closing words, and we'll shut the stream down.
No, I think not. Just to pray.
Well, why don't we just pray now?
Yeah.
I'll lead us in prayer. Heavenly Father, I'm going through stuff. Cindy definitely is going through stuff. I lift up her physical safety. I'm worried about some of those people at Dinosaur Adventureland that wish her harm. And one has been proven guilty in civil court of inflicting harm on Cindy's property. And we know that the same individual, an associate of Kent Hovind, has threatened her life. And so Lord, it seems every day I'm just glad to hear that, I get an email or something, or we meet on the internet and I know that she's okay, that she hasn't been harmed. I know I lift up her financial situation. Lord, I lift up Schnoil, who said his mother is dealing with some possible neurological disorders or something else. Hopefully it's something that's not serious. And he wrote me and asked specifically for prayer, and I lift him up. Lord, I lift my business up because I need income so that I could have more time to do shows like this and also help me with my day job. And Lord, there are just so many needs. Lord, we live in such a troubled world. You told us to pray for our leaders, to offer thanks for all men, and that it would include our terrible president and our evil vice president and our Congress. But you've said to offer thanks for them. And I can thank you on this basis that you work all things for the good of those who love you. So even though, Lord, you appointed an evil Pharaoh over Egypt and you hardened his heart, you had a plan in mind that your power and glory would be manifest through using them. And Lord, we just trust you that you will work all things for the good of those who love you. And that neither death nor life, nor things present nor things to come, neither height nor depth nor any other creature in all creation will be able to separate us from the love in Christ Jesus. And with this in mind, help us to move forward with courage and to face each day and each hour and the challenges of life. We offer this prayer in the blessed and matchless name of Jesus Christ, our Lord and Savior. Amen.
Amen. Thank you, Sal.
And thank you all. Ben Rex, it is great to see you and hang out with you. And I don't know the answer to your question here, but Lord willing, we'll talk again. Cindy, I'll meet you backstage for a little bit. And good night, all. Have a great Saturday.
God bless you guys.
Amen.